Hi, my name is María José Tárrega, and this work is part of my industrial PhD research conducted jointly at the Universitat Politécnica de Valencia y Global Omnium Medio Ambiente. In this study, we compare 12 anaerobic digestion models with a very specific objective to evaluate which modeling approach is the most suitable for predicting methane production. And it ends with a simple question. Which model should we use when we want to predict methane production in an anaerobic digester? To understand why we asked this question, we first need to put the problem in context. Europe produces more than 216 million tons of pig manure every year, while wastewater treatment plants account for around 2% of European electricity consumption. In this context, anaerobic digestion offers an attractive opportunity to recover energy and improve the sustainability of wastewater treatment plants. However, methane production is highly complex. Therefore, this work had two main objectives. First, to compare three modeling approaches in terms of predictive accuracy and computational cost. And second, to identify the most suitable models for future digital twin applications. Before comparing the models, it was essential to build a dataset that was both comprehensive and representative of real operation conditions. To achieve this, we operated 60 mesophilic anaerobic digesters for 100 days under different feeding strategies. We used two different inocula, one previously acclimated to pig manure and another with non-prior exposure to this substrate. Importantly, the models were trained using data from the non-acclimated digesters and validated using the acclimated digesters. In other words, we were not only interested in identifying which model best fitted the data, but also which one maintained its predictive capability when operating conditions change. Next, I will present the results obtained with the 5 ADM1 based models and discuss how each of them fits the different experimental dataset. We begin with the standard ADM1. This model represents the classical biochemical stages of anaerobic digestion. In terms of performance, it works very well for the A control digester, which was fed only with wastewater sludge. It performed poorly in the manure-fed digesters. ADM-B1 introduced syntrophic acetate oxidation. This alternative pathway allows acetate degradation when acetoclastic methanogens are inhibited by elevated ammonia levels. As a result, their model showed a significant improvement in the manure-fed digesters. ADM-B2 incorporates microbial adaptation by modifying kinetic parameters according to operational history. ADM-B3 focuses especially on free ammonia inhibition and the effect of the elevated pH. And ADM-B4 combines all these mechanisms into a single framework, including SAW, advanced ammonia inhibition, and solid retention effects. From a theoretical perspective, this should be the most complete model for the family. However, its very high computational cost and extensive calibration requirements limit its practical advantages over data-driven approaches. Next, we move to the second family of models, machine learning approaches. Unlike ADM1 based models, these algorithms do not attempt to explicitly describe the biochemistry of the process. Instead, they learn directly from the data. We begin with supervector regression with researchers for nonlinear relationship between operational variables and methane production. This model successfully identifies general process patterns, although it shows a limited extrapolation capability when operating conditions change significantly. The second model was a random forest. It used measurements such as ammonia concentration and organic loading rate. This model gave good results among the tested methods. However, some evidence of overfitting was observed in the most complex digesters. XGBoost achieved the best overall performance within this family, especially for a system with a complex behavior. We also evaluated a classical neural network, which produced good results, but not also significantly below those obtained with XGBoost. This was likely due to the relative limited size of the dataset. Finally, we tested LSTM, a neural network architecture specially designed for time series analysis. This model performed particularly well in acclimated manual-fed digesters, as it was able to capture the biological memory inherent in anaerobic digestion. Overall, the results show that data-driven models can successfully reproduce complex process behavior without needing to explicitly represent the underlying biological mechanism. Finally, we evaluated the third family of models, the simplified or semi-empirical model. The first was the modified Comper model. This model achieved the best results of the interstate, reaching R-squared values close to 0.99 in almost all digesters. This occurs because cumulative methane production naturally follows a sigmoidal pattern. The second model was the EV-based model. This approach also showed excellent performance with an almost negligible computational cost. This finding led us to a very interesting conclusion. After evaluating all 12 models, this figure summarizes the main outcome of the study. When predictive accuracy and computational cost are analyzed simultaneously, three clearly differentiated regions emerge. ADM1-based mechanistic models provide the highest level of process understanding. Machine learning models offer an attractive compromise between predictive performance and computational effort. However, the most surprising finding is the so-called efficiency zone. Both the models simplified achieve predictive accuracies above 0.97. Therefore, our results suggest that the model selection should be driven by the final application. To conclude, let us return to the question that opened this presentation. Which model should we use to predict methane production in anaerobic digesters? Our results clearly show that there is no universally superior model. The most appropriate choice depends on the final objective of the application. If the goal is understand biochemical mechanisms, metabolic pathways, or inhibition process, ADM1-based models remain the best option. Machine learning models provide a best balance between accuracy and flexibility, and simplified models offer the best compromise between predictive performance and computational cost. Ultimately, this work demonstrates that model selection should be driven by the needs of the application rather than by mathematical complexity alone. In conclusion, there is no best model, only the best model for a given objective. Thank you for your attention.